Good afternoon, everyone. This is Chuan Wang from Fuji Xerox. In today's presentation, I'd like to introduce a work I've done in UC Davis, an interactive visual analysis tool for cellular behavior studies using large collections of microscopy videos. In this study, we are collaborating with the Department of Chemistry in UC Davis to help the scientists study cell dynamics and function using microscopes. From here, I will call this system CellVis for short. Large collections of videos are generated from controlled experiments. These experiments are conducted under different stimuli and experimental conditions. Scientists try to understand cellular behavior against these stimuli by observing time-evolving cellular features, such as area, shape, and cell migration trajectory. One of the most dramatic and dynamic cell shape change is the cell blabbing, as you can see in these images. On the right side is the subregion of an example video. It shows the very beginning of an experiment recorded right after a stimulus to the cell. It's 30 times accelerated. Experiments like these can last for hours. For comparative studies, it's difficult for scientists to look over cell features individually and then mentally correlate them. Information visualization can become helpful by visually summarizing the multidimensional information for all the videos in a study. Great work has been done for video visualization and summarization. They can be categorized into two types. The top row shows the keyframe-based method, which trims the content of videos and emphasizes more on the presentation of featuring frames. They blend foreground motion to a 2D background or 3D time-space volume. This kind of method is less often used in scientific studies, such as in cellular behavior visualization, because scientists usually expect to see the continuously changing pattern of cell feature to be visualized and analyzed. The other category of video visualization is the abstraction-based method, as shown on the bottom row. These methods summarize attributes from video streams and visualize them. On the bottom left, Duffy's work is focusing on cell video visualization as well. They developed a glyph-based microscopy video summarization for semen analysis. The visualization conveys spatial and temporal context in one single graph. While comparing videos and videos in a quantitative illustration can be difficult, in our study, the design should be able to convey continuous measurement of multiple features simultaneously. So what kind of needs are there for cellular behavior study exactly? We interviewed domain scientists and derived these system design goals. First, the visualization should be able to summarize the feature evolution process into one single view so that scientists can quickly identify the evolving trends in features within a video. Also, scientists want to identify if the change in cellular feature is correlated to the change in some experimental parameters. Therefore, CellVis should allow easy comparison of the visual summaries across a collection of videos. Last, scientists usually need to arrange the data in a certain way to verify their hypothesis. They may also want to explore more possibilities to discover new patterns. So it is crucial to support interactive operations for data manipulation, such as sorting and filtering. This is the system flowchart of CellVis. It contains two modules, the data pre-processing module on the left-hand side and the visualization module on the right. We applied computer vision techniques to recognize cells in each frame and track them across all the frames in each video. For the videos with multiple cells recorded at the same time, we assign the cell of interest according to its location, shape, and size. We also provide a verification interface so that the user have the option to choose another cell for every video. Based on the detected cell contour, we can quantify features listed here. We also have the parameters used in the cellular experiments. Both the quantified features and the parameters are saved into files and become the input of the visualization module. Scientists then explore this data with the visualization module, which contains a viewing panel and a floating control panel. The viewing panel displays the visualization in a spreadsheet fashion. Each cell in the spreadsheet is a summarization of the change in one feature within one video. By the operations on the control panel, scientists are able to explore the feature summarization in different aspects by reorganizing the spreadsheet cells. Now let's take a look at the visualization of each cellular feature in detail. We visualize one-dimensional features such as area and perimeter in a radial bar chart fashion rather than presenting it horizontally. In this way, the radial layout is more compact. It saves more screen space when putting multiple visualizations side by side for comparison. The other reason is it's easier to control the visualization size to be the same if we normalize the timeline by setting the longest video to form a full circle. This example shows a visual summary of the area change over the first 50 frames in the video with this file name. Each area value in one time step is represented by the height of a bar, and the bars are arranged in clockwise order starting from 12 o'clock, from the start frame to the end. The area values for all cells are normalized to a range that's decided by domain scientists. On the top right corner, we show the experimental parameters used in this video. Since some findings are not published yet, we use a generic terms to pair it to represent the real parameters. From this example, we can see that the area is decreasing dramatically during the first half of the 50 frames and keep about the same during the second half. For the visualization of blobs, we also adopt the radial bar chart. The difference here is we use the height of a bar to represent the number of blobs at a certain time point, and the size of each blob is presented by color. Each bar is segmented into equally sized sticks, and each stick represents one blob. The color of the stick then encodes the size of the blob, where a darker color is used to refer to a bigger blob size. As the example shown here, at the beginning of the 50 frames, there are fewer but bigger blobs. As time proceeds, bigger blobs retract while more amount of smaller blobs appear. We plot cell migration trajectories onto a 2D Cartesian coordinate system. We set the cell centroid of the first video frame to be the origin. The cell centroids in the later frames are drawn as dots located at their relative positions to the first centroid. The color of the dots encodes time information, where light blue refers to the beginning of the video, and as time proceeds, the color of the dots become darker and darker. This example shows that the cell stays around the original location for a little while, and then it starts to migrate towards its left after a certain point of time. To allow scientists to explore their video data in different perspectives, Cellular provides several data manipulations that would reorganize the layout in viewing panel. I'd like to show this video demo. When exploring all the features with Cellular, user can scroll vertically to view different cell features and horizontally to view different videos. User can also focus on a single feature, for example, bleb number and size. By shrinking the time window size, user can magnify part of the experiment and the bar chart will expand clockwise. Then it's possible to analyze different blabbing patterns for a specific time range. In cell migration trajectory analysis, sliding the time bar back and forth will give a straightforward idea on the migration directions and distance. User will be able to select different start time to view migration stages and review the relative centroid positions to the selected start time. Clicking one of the experimental parameters will enable the sorting by that parameter. To efficiently use the screen space, user could also adjust the visualization size using the floating control panel. Now let's take a look at the case studies. These figures show the visualization of 3 blabbing cells. They exhibit the most visually different behavior. The visualized data suggests that the magnitude of the applied stimuli correlates with the blabbing behavior directly. From figure A and figure B, we can see that cell B forms bigger and more number of blabs during the whole process compared to cell A, which was triggered with 2.2 times lower stimulus. It was found that the larger magnitude of stimulus 2 trigger more intense continuous blab formation. Further comparison shows that another type of stimulus, as marked parameter 1, significantly triggers larger blabs compared to A and B. The chart also gave an insight to the dynamics of blabbing behavior, showing that while the blab size significantly increased, the number of blabs remained unchanged. This shows that visualization allows for comparison of cell behavior and reveals trends and patterns among cells which were subject to different stimuli levels. They changed to a spherical shape. In contrast, from the second row we can see a cell with a stimulus applied on the periphery shows increased cell mobility without any significant change in cell shape and size. The scientists agree that the plots of cell trajectory give them a straightforward visual summary of the direction and extent of cells' locomotion. Moreover, they conclude that ranging the visualization of the 3 features together allows them to describe and summarize all the captured dynamic events. This case study provides a comparison of the cell trajectories where experiments were conducted under 2 different temperature conditions, the room temperature on the top and the 37 degrees Celsius on the bottom. The first row from left to right shows that a gradually increasing stimulus can activate the cellular mobility in room temperature experiments. The second row shows the experiments carried out in a 37 degrees Celsius with the similar stimulus magnitude. We can see that the cells exhibit a higher degree of the cell mobility compared to room temperature, regardless of the magnitude of the stimulus. And this mobility cannot be increased further. The data collected at 37 degrees Celsius suggests that either minimum level stimulus causes maximum activation of cellular mobility, or the cells are already activated at the physical conditions and their activity cannot be further increased. The visualized data point out that further control experiments has to be done in order to find out the relationship between the temperature and activation of the cell mobility. Cell visits can also help scientists promptly identify if a cell is alive or dead. Apoptotic cells can be in different stages of programmed cell death, and they often have similar morphology to dead cells. However, apoptotic cells still have function and can recover from apoptosis. Differentiate between live and dead cells requires longer observation of suspicious cells. These figures show that by viewing the visualization of cell trajectory and area, scientists are able to identify dead cells very quickly. Dead cells has negligible changes in cell area and mobility compared to healthy or apoptic cells. The scientists also noticed that the visualization can potentially be used to track the cell viability if specific viability tests cannot be carried out. In conclusion, CellVis can summarize the multidimensional information extracted from a large collection of videos. It enables scientists to study dynamics of cellular features change over time. Also, CellVis allows quick data review and hypothesis verification via comparison quantitatively and qualitatively. We achieved the design goals mentioned earlier, and the 5 case studies conducted together with scientists demonstrate the effectiveness of our design. Right now, we are working on adding more features to CellVis, such as the roundness and cell symmetry. And this work can be potentially extended to visualize other types of videos or other types of multidimensional data for comparison studies. Thank you for all your attention.